Imagine trying to track down a single suspect, not in a crowded room, but inside a massive, bustling city that literally never sleeps. Well, that's exactly what cognitive neuroscientists do every single day. They are trying to solve a seemingly impossible, deeply internal mystery, figuring out exactly how and where human cognition happens. Welcome to our explainer on decoding the brain, where we're going to look at exactly how scientists map our thoughts.
So how do scientists actually know what your brain is doing right this exact second? You might think, hey, just put someone in an fMRI scanner, ask them to read a word, and see what lights up, right? Actually, it's not that simple. To find out how our brain processes a specific thought, we can't just take a picture, because our minds are never truly quiet. Your brain is a non-stop engine of activity.
Think about it. You've got roughly 100 billion neurons up there. And right now, vast numbers of them are constantly firing just to keep you alive, keep you breathing, maintain your posture, and process everything around you. Your baseline brain activity is like a glowing, incredibly busy pastel city. It's so chaotic that finding a single suspect, like the exact brain region responsible for simply reading a word, amidst all that crazy background traffic is a logistical nightmare. So, to find clues in all this chaos, our neurodetectives need to reach for their very first specialized tool, cognitive subtraction.
Because your brain is always fundamentally on, functional imaging has to look for relative changes. The logic of cognitive subtraction is beautifully simple. If we take a complex experimental task and literally subtract the brain activity of a slightly simpler baseline task, the leftover activity, well, that should reveal our culprit.
Let's look at a classic, groundbreaking study from 1988 by Peterson and his team to see this in action. They wanted to know exactly what part of the brain recognizes written words. So for the experimental task, they had someone passively view the word cake. But remember, basic visual processing is happening there too. So for the baseline task, they had the person just look at a simple fixation cross. Since both tasks involve basic vision, subtracting the cross scan from the cake scan cancels out all that background visual noise. And boom, the remaining activity pinpoints the exact region specialized just for visual word recognition. It's a really neat mathematical trick applied straight to human biology.
But, every good detective story has a complication. And this first tool has a pretty significant flaw known as the pure insertion problem.
Here's the issue. Cognitive subtraction relies on one massive assumption, that adding a brand new task component, like reading a word instead of just staring at a cross, doesn't change how the rest of the brain's baseline network operates. But reality check? Our brains aren't just simple addition and subtraction calculators. Adding a complex reading task might completely rewire how your underlying visual processing behaves in that moment. It's kind of like adding a really overpowering spice to a soup. It doesn't just sit on top, it completely interacts with and changes the entire flavor profile of the dish.
To get around this flaw and refine the map, scientists had to upgrade their toolkit with something called cognitive conjunction. Because that spice in the soup interaction makes imaging data ambiguous, researchers needed a way to double-check their work. Instead of relying on just one subtraction, they overlap multiple, completely different pairs of tasks that all share one common element, say tactile sensation or a specific motor movement. By looking at where these multiple different subtractions overlap, they can pinpoint the true brain area with way higher confidence. Since the weird interaction effects will be totally different for each distinct pair of tasks, finding that shared overlap effectively minimizes the pure insertion flaw.
Okay, so we've got a much better map. But right when we think we've cracked the case, we hit the ultimate plot twist in brain imaging: correlation versus causation.
This is a massive hurdle in neuroscience. The fundamental problem with correlations is that they don't, in themselves, prove causality. Functional imaging tools like fMRI or PET scans are absolutely incredible, but they essentially just reveal associations. So just because a brain area lights up on a scan when you read the word cake, that does not automatically mean that specific area is doing the heavy lifting of reading. It might just be a bystander. The brain could just be unnecessarily monitoring the situation or reacting to something else entirely. We found a correlation, sure, but we haven't proven causality.
That begs the question, if seeing a brain region light up isn't enough to prove guilt, how do we definitively demonstrate that a specific area is absolutely necessary for a thought process? In the past, we had to rely on studying patients who tragically suffered localized brain damage or lesions, but obviously we can't control those injuries, and comparing a patient to a healthy individual is incredibly complex.
To answer this in a controlled scientific way, we need a smoking gun. We need to move from just passively observing the brain to actively perturbing it using TMS. Enter transcranial magnetic stimulation, or TMS for short. This is the ultimate tool in the neurodetective's kit. It allows us to safely and temporarily turn off or inhibit a highly specific part of the brain. It literally creates what we call a virtual lesion. This gives researchers the incredible power to establish direct cause and effect without causing any permanent damage.
The way this works is actually mind-blowing. It's based on Michael Faraday's 1831 theory of electromagnetic induction. Basically, a huge electrical current rapidly flows through a figure-8 copper coil, which creates a fast, time-varying magnetic field. When held to the scalp, a massive submillisecond magnetic pulse safely passes right through the skull. It induces a tiny electrical current in the brain, temporarily scrambling the electrical processing in an area of just about 1 cubic centimeter.
So how do researchers actually use this to solve the mystery of causation? It's a three-step process. First, using an fMRI, they identify the suspect brain area. Second, they apply that harmless TMS magnetic pulse right while the participant is in the middle of doing a cognitive task. And third, they measure the change in behavior to see if the person's task performance drops.
And here is where the magic happens. If applying TMS to that specific suspect area causes your performance on, say, a reading task to suddenly plummet, we have a massive breakthrough. We've officially moved past mere correlation. Because if disrupting the area actively disrupts the behavior, we can strongly conclude that exact brain area is absolutely necessary for the cognitive process. We've proven causation.
Case closed indeed. Let's quickly wrap up by summarizing this incredible neurodetective toolkit. When you combine these methodologies, you get something truly profound. Scientists use cognitive subtraction to get their initial leads. Then, they use cognitive conjunction to refine those leads and cross-reference their maps. And finally, they bring in TMS to step in, perturb the system, and prove actual definitive causation. Together, these tools allow us to navigate that crazy 100 billion neuron pastel city of our minds to isolate exactly how and where human cognition actually happens. It is absolutely a remarkable time for neuroscience. As our tools for decoding the human brain get sharper and more precise every single day, we're literally mapping the architecture of our own consciousness. And honestly, it leaves us wondering, as this technology advances, what undiscovered mysteries, what deeply hidden mechanics of human thought are still waiting inside your own head right now? Thanks so much for joining me on this explainer, and keep questioning the incredible world and the brain around you.